figure out how to fix the ground?
I did not, so I
It's
just.
going. Right, you guys, we're moving on. We talked about citric acid cycle, okay? We're gonna start getting into fatty acid metabolism, okay? So again, a catabolic reaction. Remember, catabolic reactions are the reactions that break down the mass. When we say fatty acid metabolism, it's like the breaking down of our fatty acids. Okay? So there is one more assignment in your workbook that, depending on where we end today, I started, but it's section 33, so the acid cycle. I'm gonna assign it like a homework in your Canvas. I'll make that adjustment. But I think it's a useful exercise to do. Okay. And like I said, if we have time, we'll do it together. Or at least start it today. But we are gonna move on to the fatty acids. Okay. So we just got done looking at the citric acid cycle, okay? And we saw that acetyl-CoA, right, feeds into citric acid cycle, 2-carbon molecule, right, combines with 4-carbon oxaloacetate. And it formed a 6-carbon citrate. There was 2 oxidative decarboxylations where we lost 2 carbons and we got back to 4-carbon oxaloacetate, okay? We said that the formation of oxaloacetate was not a favorable reaction, so the concentration of oxaloacetate tends to be low. Okay. We've also seen that oxaloacetate, right, can get pulled off for gluconeogenesis, right? We've seen we go from pyruvate to oxaloacetate to PEP, and then it keeps going to make glucose. Okay, we've seen that. Okay. Now, in our discussion so far, that acetyl-CoA came from pyruvate. Okay. We saw pyruvate goes with pyruvate dehydrogenase complex, makes that acetyl-CoA. But we said at the very beginning that acetyl-CoA could come from fatty acids. Okay, and that process is beta oxidation. That is fatty acid metabolism. Okay, and so I want us to think of a scenario. Okay. Either your starvation, okay, or a very low calorie diet, okay, and in that sense, there's no source of glucose, okay, or if you have untreated diabetes, okay, and remember in diabetics, what's happening is you there is glucose in the blood. But your cells are not aware that the glucose is in the blood. They don't know they can take it up and use it. Okay. So that's why they got to insert that insulin, remember, that signal to the body, hey, there's a lot of glucose in the blood, take it up. Okay, that's why diabetics gotta do those insulin shots. So in untreated diabetes, they have low insulin levels, so the tissues do not take up glucose from the blood. So it's almost, they're like in this. The cells think they're starving, even though it's there, they don't know anything. Okay, so it's similar. So then what happens in the body, when that happens is you start using your citric acid cycle intermediates, like that oxaloacetate. You start using that up in G, which is gluconeogenesis, right, for all the alphabet soup here, right, C, G, and G. Okay, you start using up in gluconeogenesis. And remember that oxaloacetate is already in low concentration. Okay. So the acetyl-CoA that's coming from the breakdown of fats, it's not going to have very much oxaloacetate to combine with because it's getting pulled off for gluconeogenesis. Okay. So then those acetyl-CoAs have no citric acid cycle to feed into. So what happens, that concentration of acetyl-CoA starts to build up because citric acid cycle intermediates are building up. Okay. And again, this comes at the end of chapter 17, but I found it helpful to bring it up at the beginning. So when we get to the end, it'll make even more sense. Okay. So you're keeping this in mind when we're thinking about the breakdown of fats. So what happens is that those acetyl-CoA molecules, they start to combine with each other. Those two carbon molecules, they start to combine with each other, and that is what makes ketone bodies. And we'll look at the structures of those and what that how they combine, but this is just introduction to this idea, okay? They start making ketone bodies. And ketone bodies are this acetoacetate, this D-beta hydroxybutyrate. Okay, those are all ketone bodies. The concentration of those start to increase, and that leads to that condition known as ketosis. Okay. So those ketone bodies lead to ketosis. At the same time, they lower the blood pH. Remember, the pH scale is on the logarithmic scale. Okay. So it goes from like 7.4 to like 7.2, but that's like leads to a person being in a coma. Okay, because it's a huge change in pH for the blood. It's very sensitive to those changes. So that low blood pH, that's the acidosis. So I think you guys might have heard of ketoacidosis, untreated diabetes. My son's reading a book right now. The little boy is in a diabetic, and the mom passed away from ketoacidosis. And they're like, "It's not really happening." I guess so. I mean, like that's what I say in class all the time. I say it can lead to coma or death, but I don't know. Is anyone working in a medical field? Oh, yeah, that's right. Or yeah, you guys ever see that or anything? Anyone in ketoacidosis? Sometimes I have students in this class that have seen it. I personally have not seen it. But this is what leads to that condition, that ketoacidosis. Okay, and so it can get dangerous. And so again, the body thinks it's starving, it starts breaking down fats, but then those fats lead to acetyl-CoA that combine to make those ketone bodies. Yeah, they look
Kind of unrelated, but related. How does it like the keto diet, how you're like in ketosis, like how does that work?
Well, so there, right, in keto diet, you're eating a lot of protein, right? And so you don't have a source of carbohydrates. So you do have like low, like there's not a source of glucose. Huh?
Yeah, it's like
A lot of fats, right? There's a lot of fats. So there is some research showing that a certain amount of ketosis could be actually beneficial, but I think that people that are on hardcore keto diets are supposed to monitor their
short-term diet.
Yeah.
Yeah, and I also heard it's like long-term is bad for like cardiac health and stuff like that. Like
Yeah, well, again, because you're getting this high fat content, right? Which is probably again, I think research is changing, right? Because fats are not all that bad, right? People used to be like, oh my gosh, low fat, but I think the verdict's out that we need fat in our diet to help us absorb certain nutrients and that it helps us feel fuller and all those things. But again, keto diet is do at this at a like this is the extreme, right? This coma or death, right? Keto diet's like a less drastic version of what's going on here. Okay. So again, you're not taking in sources of carbohydrates, right? You're taking in a lot of protein. So then you're burning your fat as a source of energy. Okay. And then yes, you're gonna get a certain amount of ketosis because you don't have an immediate source of glucose all the time. Okay. But then I know sometimes people are monitoring there's some way to measure your ketone bodies or something. Okay. Okay, so that's like in words, right? But here's the rest of that figure, right? We're pulling off oxaloacetate from citric acid cycle to go through gluconeogenesis to make glucose, right? In the liver, the glucose gets exported as fuel for the brain and other tissues, okay? But then that means this acetyl-CoA doesn't have that oxaloacetate. If a lot of it is getting pulled this way, then there's less of this to combine with acetyl-CoA. Okay. So acetyl-CoA, again, it goes this way instead. It starts to combine with each other, and it leads to that ketone body formation. Again, the ketone bodies are acetoacetate, D-beta hydroxybutyrate, acetone. They get exported as energy sources for the heart and muscle, kidney, and brain. So your body can use those ketone bodies as a source of energy. They'll take them in, they'll break them back down to acetyl-CoA, and they can use them in their citric acid cycle. Okay, that's why the body does this ketone body formation in the first place. Okay, it's like a way to maintain homeostasis even if we're not taking in glucose. Okay. So they are a source of fuel. It's just that they lower our blood pH. So if we get too high, it leads to dangerous conditions. Okay. So I like to bring that to mind when we're thinking about the breakdown of fats again, and this is an untreated diabetes or a starvation. Okay. Now before to understand how fats get broken down, first we got to think about how we digest fats. And there's some vocab I like to review because I just want to make sure we're all on the same page and remember stuff from CHEM. Okay, and maybe somewhere in biology. Okay, so first term here, fatty acid. What is a fatty acid? Okay, so remember, a fatty acid has that carboxy head group. Okay, this carboxyl group on one end. Okay, and then it has just a long chain of carbons and hydrogens, a hydrocarbon chain. On the left, we have a saturated fatty acid, right? There's no, except for the carbonyl, right? In the hydrocarbon chain, there's no double bonds. Okay, so it's completely saturated with hydrogen. It's all single bonds. On the right, we have this double bond. It's unsaturated fatty acid. Okay, notice it has that, it leads to a kink in the formation. Okay, this is a cis double bond, which is what fats tend to make. We will see a trans double bond in beta oxidation, but in our body they tend to be cis. Okay. And they tend to be cis. I mean that this leads to lower melting points. Okay, which is good. They tend to be liquid. Okay, usually you want you can think about unsaturated fats. You usually want that over saturated fats. Usually trying to avoid saturated fats. And you also usually try to avoid trans fats, right? Usually in people's eyes, they're trying to avoid trans fats because a trans double bond would not lead to this kink, which would allow for these hydrocarbon chains to stack up and have more intermolecular interactions, which lead to higher melting points. So they're more solid in your body, which
They make them, I'm
huh?
not sure if I'm correct. They make the membranes inflexible because of,
Yes, yeah, they make membranes flexible.
I mean, whenever it's, when trans fat is taking the place of cis fat, the membranes have to be flexible, but the trans fats make them
Stiff.
stiff.
Yeah, yeah. Yeah, so
But
this
stiff
keeps
like
things
the-
more fluid,
Yeah.
the cis stereochemistry of that. Yeah. And it's because, right, it leads to this kink in the chain, so the chains can't like pack up against each other. Because imagine if you had a lot of these could just one right after another, right? Well, that's not really possible with that stereochemistry. Okay, so again, just remember the structure of a fatty acid, okay? Triacylglyceride, okay? Remember, that is a glycerol molecule, which is shown here. Okay, here's glycerol, and I always think of a glycerol as an outlet, like in the wall, but it has 3 plugs. Okay, so I think of this as like, here's 1 plug, 2 plugs, 3 plugs. And for tri-3-acylglyceride, you have plugged in 3 fatty acids into that glycerol molecule. Okay. So tri-acylglyceride is a glycerol plus fatty acid. I don't know if you guys exposed yourself to this concept, this chapter yet in 17 in Leninger, but there's all kinds of vocab and they're throwing around triacylglyceride, diacylglyceride, monoacylglyceride, right? Fatty acids. So I just want to make sure that when they're saying those things, we're thinking about what they mean. Okay. So again, this is a triacylglyceride. 3 fatty acids plugged into glycerol. And they'll talk about micelle. Okay, a micelle. So remember, a micelle is where you have, you know, that you could have a fatty acid, right? Something with a hydrophilic head and a hydrophobic tail, okay? And all those hydrophobic tails form the core of this micelle, with those hydrophobic heads on the outside towards the aqueous environment, okay? Because again, the head is hydrophilic, the tails are hydrophobic, they want to hide away from that water. And so they hide into the core of a micelle. Okay, so that's just like a cutaway, if you were to cut one in half. Kind of what it would look like. Okay. So. That was vocab review. Now we get to think about digestion. Just real quick though, we look at the triacylglyceride. Again, I said you can have mono and diglyceride as well, okay? I want you to think about when we were going through the citric acid cycle and we lost carbon dioxide. We said that was like the most oxidized form of carbon, right? The carbon had 4 bonds to oxygen, 2 double bonds to 2 oxygens, okay? Now, this is a highly reduced structure here, okay? When we look at that fatty, I don't know if you guys are seeing it that way, but this is highly reduced. There's lots of electrons stored in here, okay? So triacylglycerides are really suitable for storage, okay? They're highly reduced. We're gonna see we can extract a lot of electrons out of there to make ATP. Remember, like I said, it's a flow of electrons that not only powers our electronics, but it powers us too, okay? They're not soluble in water, right? That hydrophobic tail is not soluble in water. So they don't need to be like dissolved in water, right? They can be stored in the adipose tissue as adipose droplets, okay? So that's better for storage. Imagine if all our fat had to be dissolved in water, how much more weight we would carry around, okay? They're chemically inert, right? This is not, when you think of chemistry, I mean, maybe at the carbonyl, but right, this carbon chain is not something you really think of as very chemically reactive, okay? And we're gonna have to overcome that in order to use it as fuel, okay? So we're gonna have this this is a great way, this is how we store our energy away, okay? And we need to overcome that unreactiveness to use it as a fuel, okay? All right. So we tend to have 3 sources of fats, okay? From our diet, so the food that we take in, okay? We have fat stored away in our adipose tissue. And we have fats that are being synthesized because of, we have excess energy. We are meeting and exceeding our energy demands, and so we make fats and we we can export them to the adipose tissue, okay? So dietary fats, when you take them into your diet, get absorbed in the small intestine, okay? So where are we? We're in the small intestine. What's happening here? Digestion of our dietary fat, okay? I'm not a nutritionist. I always feel funny covering this, but it's interesting. It's helpful to think about as far as like what our body is using as fuel. Preferentially, it's always glucose, right? Fats take a little bit more work. Why? We're gonna see, because this is how we mobilize our fats to use for energy, okay? So again, digestion of the fats begins in the small intestine, okay? This is where cholesterol is important, okay? Cholesterol makes bile salts. And bile salts have a non-polar tail with a polar head group. So these bile salts, okay, can emulsify dietary fats in the small intestine, forming mixed micelles, okay? And so you also have lipases. Do you guys feel comfortable with this word, lipases? So in like molecular biology, biochemistry, whenever you have that ASC, like there's RNases, there's proteases, DNases. They're breaking down, right? This is an enzyme that's breaking down fats, lipids. Okay, DNases are enzymes that break down DNA, RNases are enzymes that break down RNA, proteases are enzymes that break down proteins. Okay. You have lipases in the small intestine, and they're going to degrade those triacylglycerols. So they break it down, and now that they're a little bit smaller, they're not 3 fatty acids tucked into glycerol. They can be taken up through the small intestine, okay, the intestinal mucosa, okay, so they can pass that lining. And then they get converted back to triacylglycerols. Okay, they get converted back. And then along with cholesterol and other lipoproteins, they form these chylomicrons. Okay, so this has triacylglycerols incorporated with cholesterol and apolipoproteins. Here's a look. We'll come back to this slide, but just so you see those chylomicrons. Okay, apolipoproteins, triacylglycerols, and cholesterol form these big. Not big, but again, these are how lipids get transported in the blood because they're not water soluble, right? They're not water soluble. So this is how they get transported in the aqueous environment like our blood. Okay. But look at that close up. That looks so weird. Little triglycerides in there. Okay. So they can move through the lymphatic system and bloodstream to tissues. Okay. Now you have a lipoprotein lipase. Okay. And it can convert the triacylglycerols back to fatty acids and glycerol. Why? Why does it do that? Because it's going to have to cross another barrier. Okay. It's going to have to cross out of the bloodstream, and it can either go to an adipocyte, right? A fat cell for storage. Okay. Or it can go to a myocyte where it's going to get oxidized as fuel, right, to form carbon dioxide and ATP. Okay. So notice it's like it's kind of weird. It goes from triacylglyceride, they break it down, cross the barrier, form the triacylglyceride again, travel through the bloodstream, break it down again. But it's like every time it gets broken down, so it can cross, you know, either the small intestinal lining or from the bloodstream into a cell, into an organ. Okay. 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 Questions about that one? This is facts we take in through our diet. Okay. All right. Now, if we're looking at our stored fats, okay, hormones are going to trigger mobilization of stored triacylglycerols. Okay, hormones. So where are we? Now we're in the adipose tissue. Okay, so our fat tissues. What's happening? We're mobilizing the fats that we've stored away. We're saying, come on, get out of storage. It's time to go burn you as fuel. Okay. Let's see what's happening here. Okay. Look at, we have glucagon binding to a cell surface receptor, leading to activation of this G protein, right? That leads to cyclic AMP, right? From ATP, which activates PKA, protein kinase A, okay? Which is leading to phosphorylations of these things called these perilipins, okay? Maybe perilipins. Have we seen anything like this before? A series of events. Yeah, we have. Yeah. Like you're very dramatic, but yes. Okay. Yes. Yeah. These cascade signals, right? We saw it specifically with glycogen breakdown and synthesis. Okay. But remember, that's kind of the power of those hormonal signals, right? Glucagon is doing many things. We saw kind of what it was doing at the liver. Look what it's doing at the adipose tissue. Okay, it's leading to this mobilization of fats. Okay, so what's happening here? So you have this lipid droplet, okay, and the surface of the lipid droplet is coated with these perilipins, okay, and they kind of restrict access into the fat stored in the lipid droplet. Okay. But if you get a glucagon signal, again, that's a signal that we like that there's a demand for energy, right? Glucose is gone, we need some energy. Okay. Again, it goes through this series of events with adenylyl cyclase, which leads to cyclic AMP, activation of protein kinase A. And protein kinase A is a kinase. It comes and phosphorylates these perilipins, and that causes them to open up. Okay. It causes the lipid droplet to open up, and that allows the free fatty acids stored in there to get released. And the free fatty acids are released into the blood. Okay. Now, again, these are fatty acids. They get carried in the blood by an important protein, serum albumin. Some of you are working in 441 with BSA, right? Bovine serum albumin. You maybe heard of that in some other bio class, BSA. BSA always comes up. Bovine serum albumin. What does it do? Right? Well, that's what that protein is for. Is we're carrying fatty acids in the bloodstream. Okay, but it's used as a standard kind of protein in a lot of molecular biology, biochem applications. Okay. So the serum albumin can carry the fatty acids to the blood, and they can take it to like muscles, hearts, okay, heart tissue. And then that gets moved into the cell with a fatty acid transporter, where it can go through beta oxidation, which leads to citric acid cycle and respiratory chain for a lot of ATP and carbon dioxide. Remember that fatty acid also can have a glycerol portion, okay? That glycerol portion can feed into glycolysis. There's a method for glycerol to feed into glycolysis as well, okay? All right. Questions on this one? This is mobilization of our fat storage, right? Of the fat stored in our adipose tissue. Okay. Yeah, so we have seen this hormonal cascade before, like, and I kind of, that's the one. Okay. All right. Let's look at the structure of mitochondria. Okay. We've talked about mitochondria a little bit. We've seen pyruvate get transported through the mitochondria when we looked at gluconeogenesis. And we'll start talking about the mitochondria more and more when we get into electron transport chain and oxidative phosphorylation, but it's important for fats as well, okay? So just a reminder, maybe you guys already know this from some other class, but just so we're all on the same page, right? The mitochondria has an outer membrane, okay? The outer membrane has these pores that are relatively large, and they're freely, that allows, you know, small molecules and ions to freely pass through these pores of the outer membrane of the mitochondria, okay? But the mitochondria also has this inner membrane, okay? And this inner membrane is this one that has all these folds, the cristae, okay? The more folds you have, the more surface area you can have. And that's where on the surface of this inner membrane, okay, it contains your electron chain, right? Complexes 1, 2, 3, and 4 are located on the inner membrane. ADP, ATP translocase is there. ATP synthase is on there. So again, the more surface area you have in that inner membrane, the more of those enzymes you can have, okay? And there's a lot of transporters because that inner membrane is impermeable to most small molecules and ions, including protons, okay? Because remember, you got to create that proton gradient to make ATP. We'll talk more about that later, but maybe you've heard about that before. There's a proton gradient. So that inner membrane is very selective about what it's allowed to pass through it, okay? And that's important. So inside of that inner membrane is called the matrix, okay? And in the matrix, we have that pyruvate dehydrogenase complex, which we saw took pyruvate and made acetyl-CoA, okay? We have the citric acid and cycle enzymes are inside the matrix, okay? And the enzymes we need for fatty acid beta oxidation are also inside the matrix, okay? That means we've got to get our fats in there. But remember, that's a very selective membrane, okay? Also, the enzymes needed for amino acid oxidation are inside the matrix. Notice most catabolic reactions, the breaking down, right, of our proteins as amino acids, our fats, and the citric acid cycle and pyruvate are in the mitochondria. The one major catabolic process that's not in there is glycolysis. That's in the cytosol. That's happening out here somewhere. Okay, many other enzymes, DNA ribosomes, okay, are in there. Okay. Structure, questions about the structure of mitochondria. Just kind of keep it in mind. Again, we've talked about it, but now we're getting more serious now. Okay, all right. Okay. So, like I said, we got to get our fats in there because that's where the enzymes are to break down these fats. Okay. So again, our fats are degrading to fatty acids and glycerol in the cytoplasm of the adipocytes. Okay. Fatty acids are transported to other tissues for fuel. Beta oxidation of fatty acids occur in mitochondria. Small, less than 12 carbon fatty acids can diffuse across the mitochondrial membranes. But most fatty acids are larger, okay? And they have to be transported via this acyl carnitine transporter. Okay, so they have to be transported in. Okay. So we call this the carnitine shuttle. Okay. And so the first reaction here is with acyl-CoA synthetase. Ooh, synthetase. Not synthase, synthetase. What does that tell us? That extra syllable in there? It requires ATP. Okay, ATP is required. Okay. So that enzyme, right, is going to attach, it's going to lead to the attachment of a coenzyme A to our carboxyl group of the fatty acid. Let's see what's happening. Okay. So here in pink represents our fatty acid. Remember, our fatty acid is that carboxy group with this large hydrocarbon chain. So there's this big hydrocarbon chain not being shown here by that R group. Okay. ATP is involved. ATP is going to activate this fatty acid to get broken down. Okay. So we have the carboxylate ion is picking up the phosphate group from ATP, okay, and it forms this fatty acyl adenylate, okay. Notice it's attaching this part of it, like AMP, to the carboxyl group of the fatty acid, okay. So that releases this PPI, represented PPI, these two phosphate groups, pyrophosphate, okay. Remember that those can immediately break down into 2 molecules of inorganic phosphate. Now, this is a great leaving group, okay? So coenzyme A can come in and attach to the carbon of the carbonyl here, and that's going to release AMP as a leaving group, okay? And now we form this fatty acyl, referring to our fatty acid, but it's attached now to coenzyme A, this fatty acyl group, okay? Here's that pyrophosphate. Sorry, I didn't notice that on the slide there. There's that PP being released and breaking down to 2 inorganic phosphate. Okay, so that's the first reaction. Okay, so we've activated our fatty acid by attaching it to coenzyme A. We had to use ATP to do this. Okay. Okay. This first reaction is happening in the cytosol. This product can be used for membranes, if it wants to be used to make membranes, or it can be transported into the mitochondria for beta oxidation to produce ATP. Okay, it's going to depend on the needs of the cell. Okay, so if it's going to go into the mitochondria for oxidation, then the fatty acid is going to get attached to another molecule. It's going to get attached to the hydroxyl group, okay, of this molecule called carnitine. So this is the second reaction. So carnitine is going to shuttle our fatty acids into the mitochondrial matrix. Okay, so this is like our molecule that carries our fatty acids inside that, into the matrix of the mitochondria. Okay. So out here, look at, on this, picture, we have the cytosol on the left. We have that outer mitochondrial membrane represented here, okay? We have this intermembrane space, the space between the two membranes. Here's our inner mitochondrial membrane. And here's the matrix. So this is where we're trying to get our fats into, okay? We just saw how we activated our fatty acid by attaching it to coenzyme A, okay? So the fatty acid is going to get transferred to carnitine. So notice, right? Coenzyme A has been released. Our fatty acid is now attached to carnitine. Okay. That's happening in the cytosol. Okay. And it gets carried through the outer mitochondrial membrane by this carnitine acetyltransferase 1. Okay, this inner membrane, this membrane protein. Okay, this protein is part of the membrane that allows for transport into the matrix. Once it's inside the matrix, that carnitine transfers the fatty acid to coenzyme A in the matrix. Okay, so then carnitine is released, and now we have this fatty acyl CoA inside the matrix where the enzymes are there to break it down. Carnitine can come back through and pick up another fatty acid. Okay, so it's literally taking it from coenzyme A in the cytosol, transporting it through the membranes, and then delivering it to another coenzyme A in the matrix.
Is it because coenzyme A is too large?
Yes, so coenzyme A, right? You have a pool of coenzyme A in the mitochondrial matrix, and a separate pool of coenzyme A in the cytosol. It cannot pass through the membrane. So they're separate pools. That's one of the questions I think in homework and that come up on exams. Okay. So this is a rate limiting step in the breakdown of our fats, is getting them to where they actually need to be the enzymes that break them down. So this is, we'll see after we look at beta oxidation that this is a point of regulation too, that this gets highly regulated. This step.
Which point?
Just the transfer of fats into the matrix, just this whole process of getting fats into the matrix. Okay. How you guys doing? Okay, so then the next section is to start going over oxidation of fats, but I'm not going to go into that today. I'm gonna give you a little preview, okay? But like how amazing this is what happens, right? One fatty acid that gets transported into inner mitochondrial membrane, right, can be broken down through beta oxidation. And again, you cleave every 2 carbons. We're going to look at this when we come back from spring break, but we cleave every 2 carbons. And that leads to, if this is palmitate, okay, palmitate has 16 carbons, it leads to 8 acetyl-CoA that feed into the citric acid cycle, leading to a transport of 64 electrons in the form of NADH and FADH2 to the respiratory chain, okay, to make ATP. So we'll do the calculations for this later, but this is what we're gonna go into when we come back from spring break. Okay. Questions about this? The main point I think is that like it's how difficult, not how difficult, but again, it takes a little bit more work to mobilize our fats as fuel. Okay, they're not water soluble. They can't just be, their enzymes are inside the matrix. They got to get in there. Okay, and so we need that carnitine shuttle. All right. Questions for me? Melissa? right. So I'm gonna stop lecturing. What I would like for you guys to do is. Sorry, this is a little backward. Is we can get started on that, section 33, exploring the citric acid cycle. It's on page 133 of your workbook. And I think there's just some great, more fleshing out the citric acid cycle. Does anyone take a picture of my, book? Want me to picture? You can do 1 through 11. Okay, guys, let's look at this assignment together. I didn't ask you, but on the previous page on 133, if I was asked some interesting questions to think about this creating time for this exercise. It asks for the pre-activity, you know, which intermediate from the cycle is removed to make fat? No, let me picture that. So which intermediate from the citric acid cycle is removed to make fat? And we can see here, right, that citrate is the one that's removed to make fatty acids. Okay. On the pre-activity, I'd ask which intermediate is removed to make glucose. So which intermediate is removed to make glucose? What do you guys think? Oxaloacetate, right? Oxaloacetate gets removed to make glucose. And then which intermediate is removed to make glutamate? Alpha ketoglutarate. Okay, alpha ketoglutarate gets removed to make glutamate. Okay, where do fats enter the cycle? Ooh, where do fats enter the cycle?
I think at the beginning of the
Acetyl-CoA, right? They enter here. Fats into the cycle as acetyl-CoA. Okay. Okay. And so thinking about those points, right? Oxaloacetate gets pulled off for glucose. This gets pulled off to make fats. Here's where fats enter. This can get pulled off for glutamate. Okay. So thinking about those, right? Like we said, the citric acid cycle is a hub for lots of other things in our metabolism. So when you look at the delta G values for the reaction to the citric acid cycle, what are the far from equilibrium and near equilibrium reactions in the citric acid cycle? What were the far? Let's look at a different picture. What were the far from equilibrium reactions? Which one was it?
Acetyl-CoA, the number one.
Okay, so acetyl-CoA combined with oxaloacetate to form citrate. Okay. There. What else?
Number
Pyruvate to acetyl-CoA. So the kind of the pre-reaction. Yeah, and we saw regulation of that one. Okay. Number 3, isocitrate to alpha ketoglutarate. Okay. Any other ones?
Number 4.
Number 4, alpha ketoglutarate to succinyl-CoA.
Is there a specific definition for this, or is it broad? Because I don't really understand what qualifies as far from equilibrium and what
Yeah.
close to equilibrium.
So close to, like the smaller, like the closer you are to 0, right? The closer you are to equilibrium, right? The farther away from 0. So like usually you get like 20 and above, you know, it's like you'll start getting far, you know. And we have to remember that these values given to us here are they have that dash and the knot, right? They're standard and they're transformed. So they're saying this is under standard conditions, which is different from what's actually happening in the cell. So often the actual delta Gs are often larger. In the cell. And like for the last one, for the oxaloacetate, from malonyl to oxaloacetate, we see like that's like a big positive one, right? Right, it's a big positive one, but, and then again, it has an apostrophe in the knot, so this is under standard conditions. In the cell, this reaction happens, and we can affect the equilibrium by keeping the concentration of that oxaloacetate low. And that goes back to week 1, when we were talking about delta Gs and Qs and natural log of Q, and remember all those calculations we were doing at the beginning of the semester. Okay, so that's why it becomes relevant, because we want to think if we keep the concentration of oxaloacetate low, we can drive this reaction forward. Okay. Okay. So citrate, acetate, okay, okay. So what would you guys predict as the reactions that get regulated?
2, 8, and maybe 6. Positive ones.
The positive, okay, okay. What do you guys think? What did everyone else pick that? How big were those positive ones?
Well, 6 was 0.
Okay,
That's why I said maybe. 2 was 13.3. And then 8 is the 29.7.
okay. So we want to remember that points of regulation tend to happen at reactions that are far from equilibrium. So that was back in our regulation chapter. Okay, so when the reaction's far from equilibrium, that's where they tend to be regulated. So it's kind of the other reactions. Okay, so like the citrate synthase is a point of regulation that has that large negative delta G, and that is far from equilibrium. That's identified as a point of regulation. If we go back, let's see. We looked at this, and we don't have all these details for our reactions now, but we these are all the reactions of glycolysis. And we looked at what were their KEQs, right? Under equilibrium conditions, and what were they actually at in the liver and the heart. And when there was a big difference in there, okay, you get these right, you can tell if it's near equilibrium or not. Now look at the actual, these are the actual delta Gs all the way to the right, and then these are our standard transformed delta Gs. Okay, so like hexokinase is large and negative. Okay, PFK1 is large and negative. They're even more, they're larger, more negative, right? In their actual endogenous settings. Okay. But some of these reactions, like even though their standard transformed is positive, when they get to the actual conditions, the delta G changes from positive to negative here, and it's closer to equilibrium. And so we saw the reactions that were large and away from equilibrium. Like we talked a lot about regulation of hexokinase. We talked a lot about PFK1. We didn't really talk about any of these, okay? And their actual delta Gs are all kind of, they're smaller, okay? Now we get this big old negative 17, that's pyruvate kinase. So we talked about regulation of pyruvate kinase, okay? So when we were looking at regulation, it's these enzymes where the delta Gs are far from equilibrium that are points of regulation. And that's how you can kind of predict where something's going to get regulated, okay? We're about done with class time here, but what I want, something that's interesting is to think about is that if we're looking at these points of regulation, right? Citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate, and we go back and look at where things branched off, right? We're regulating at those branch points because we want to be careful, what's the need of the cell? Do we need this intermediate to continue the citric acid cycle for energy? Or can we release this intermediate to go make fats? Or to go make alpha ketoglutarate? Or to go make glucose, right? What's the need of the cell? So that's why regulation needs to happen, right, around those branching points to make sure we're using our intermediates most efficiently for the cells. All right, good. I hope you guys have a restful and rejuvenating spring break. Maybe check out a little bit on Canvas app, you know, slides and when we get back, there's kind of, we'll finish fatty acids, metabolism, and then there's an exam, the second. Okay. All right. Go ahead.
Thank you.
Yeah.